Welcome to foodfeedinfo.com. In this video we will discuss de novo synthesis of fatty acids. This is a part of fat metabolism, and actually it's a fat anabolism. Synthesis of fat, or the formation of depurred fat. The depurred fat in the body, or triglyceride, they are synthesized in the body from fatty acyl-CoA and glycerol 3-phosphate that are converted into triglyceride. And the second method is simply the preformed glycerides, they are converted into triglycerides. So we can say the depurred fat, they are derived from the preformed glyceride. So these preformed glyceride, either they are absorbed as such from the diet, or they are synthesized, or they are available in the liver. So this can take place in most tissues, but usually it is confined mostly to the liver and adipose tissues. Fatty acyl-CoA synthesis. So it is generally considered there are usually three systems of fatty acid synthesis. The first system is highly active, and it is active in the cell cytoplasm, and it results mainly in the production of palmitic acid or palmitate from acetyl-CoA, or in some cases it results in the production of butyryl coenzyme A. So almost all other fatty acids, they are produced by the modification of this acid. So this system which is active in the cytoplasm, it result in the production of palmitate, and other fatty acids, they are produced or derived from the palmitate. Then is the second system of fatty acid synthesis. The second system it occurs mainly in the endoplasmic reticulum, but to a minor extent it occurs in the mitochondria. So this system involves the elongation of fatty acid chain by the addition of 2 carbon atoms. So it means a 12 carbon atom is then converted into 14, and then into 16, and then into 18. So in this system the donor is a malonyl-CoA. Malonyl-CoA actually it provide the carbon atom required for the chain elongation. So 12 carbon fatty acid with the donor of malonyl-CoA is converted into 14 carbon fatty acid, then into 16 carbon fatty acid, then into 18 carbon fatty acid. Now look at the third system of fatty acid synthesis. So this system occur only in the endoplasmic reticulum. So this system is responsible for the desaturation of preformed fatty acid. Now we will discuss the three systems one by one. So with the first system, that is the synthesis of fatty acid in the cytosol. Particularly there is a production of palmitate as a starting point for the synthesis of long chain fatty acid. So as a first step, there is a requirement of acetyl coenzyme A to carry on the process, or to carry on the reactions of fatty acid synthesis. If we look at the production of acetyl-CoA in non-ruminant animals, so it is produced in the mitochondria as a result of oxidative decarboxylation of pyruvic acid. This pyruvic acid is produced from the degradation of glucose through glycolytic pathway, or through glycolysis. The other way of the production of this acetyl-CoA is through oxidative degradation of amino acid and fatty acid. So these are the two main sources of acetyl-CoA production in the mitochondria. So in the next step, this acetyl-CoA, after its production in the mitochondria, is of no use or cannot be used for the synthesis of fatty acid. So it should be transferred from cytoplasm to the mitochondria. If we look at the mitochondrial membrane, that membrane is impervious to acetyl-CoA. It means it cannot pass through the membrane, or cannot enter into the cytoplasm until it is complex with something, or it should be converted into some other compound. So it can be complex with the carnitine. This is a one way, or the other way is it should be converted into citrate before transport into the cytoplasm. So this is what is going on in case of non-ruminant animals. Whereas in case of ruminant animals, there is a production of acetate as a result of fermentation process, and uh, I will explain later. If we have to complete this step, just look at the reaction over here. The citrate is combined with the coenzyme A, that is then converted into acetyl-CoA and oxaloacetate. For this reaction, it is catalyzed by ATP citrate lyase enzyme, and there is a requirement of 1 ATP. So this is how the things are going on in case of non-ruminant animals. However, when we look at the ruminant animals, there is a direct production of acetate through fermentation process. That acetate is absorbed directly from the rumen, and it is changed into acetyl-CoA in the presence of acetyl-CoA synthetase. And this is going on in the cytoplasm. So this acetyl-CoA is the major source of acetyl-CoA as a precursor, or as a precursor for the synthesis of fat. And in ruminant animals, the ATP citrate lyase activity is greatly reduced, and the passage of mitochondrial acetyl-CoA to the cytoplasm is limited. The reason being, there is a direct production of this acetyl-CoA in the cell cytoplasm. So this is the comparison between ruminant and non-ruminant animals. So in the next step of this acetyl-CoA transfer to the cytoplasm for fatty acid synthesis that we have discussed, it should be complex with the carnitine and converted into citrate before transported into the cytoplasm. So this is these activities, they are going on in the liver, brain, kidney, lung, mammary gland, and adipose tissue. So we are discussing the synthesis of palmityl or palmitate in the cytosol. So once the acetyl-CoA in the cytoplasm, then it should be converted into malonyl coenzyme A. So this is the first step in the cytosol, where acetyl-CoA is converted into malonyl coenzyme A. This reaction require NADPH ATP bicarbonate, which is used as a source of carbon dioxide, and a mineral manganese. The overall reaction of this synthesis is the acetyl-CoA, with the help of ATP, water, and carbon dioxide, it is converted into acetyl-CoA with the production of ADP and inorganic phosphate. The enzyme required for this reaction is acetyl-CoA carboxylase, and the mineral is manganese. So this reaction is important in a case because this malonyl CoA then act as a donor for the provision of required carbon molecules for the synthesis of next even chain fatty acid, like from 12 to 14 carbon fatty acid. The important thing to note over here you can see the acetyl-CoA is a 2-carbon compound, so that is converted into 3-carbon compound malonyl CoA. So this is the advantage of this reaction, 2-carbon is in the form of 3-carbon malonyl coenzyme A. So in the synthesis of palmitate in the cytosol, the next step is the conversion of malonyl CoA to malonyl acyl carrier protein. So malonyl coenzyme may react with the acyl carrier protein. In the presence of the enzyme malonyl CoA ACP transacylase, that result in the production of malonyl ACP complex. Similarly, acetyl coenzyme A then react with the acyl carrier protein, that result in the formation of acetyl acyl carrier protein, and this reaction is catalyzed by acetyl CoA acyl carrier protein transacylase. And the overall reaction is this malonyl CoA then combine with the malonyl ACP then combine with acetyl ACP, that result in the production of butyryl ACP. So in this way, a 3-carbon malonyl CoA that react with 2-carbon acetyl CoA result in the production of a 4-carbon fatty acid in the form of butyryl ACP. So from 2 carbon now we have a 4-carbon fatty acid. So now we will look at the summary of all these reactions for the synthesis of butyryl ACP. The starting point is malonyl ACP combined with the acetyl ACP, and with the enzyme keto-acyl ACP synthase there is a production of acetoacetyl ACP, and there is a release of 1 carbon dioxide at this reaction, at this step. Then acetoacetyl ACP is converted into beta hydroxybutyryl ACP. The enzyme required for this reaction is beta ketoacyl ACP reductase. This reaction requires energy in the form of NADPH that is converted into NADP. Then beta hydroxybutyryl ACP is converted into crotonyl ACP. Enzyme required for this reaction is crotonyl ACP hydratase, and there is the production of 1 molecule of water. Then finally crotonyl ACP is converted into butyryl ACP, and enzyme required for this reaction is enoyl ACP reductase, and at this stage again NADPH is required. So overall there is a production of 3 intermediate while the production of butyryl ACP, and there is a requirement of 4 enzymes, 2 reductase, 1 synthase and 1 hydratase, and there is also a requirement of 2 NADPH for this complete conversion or complete production of this 4-carbon butyryl ACP by the combining of malonyl ACP and acetyl ACP. The reactions are taken from McDonald Basic Animal Nutrition book, highly acknowledged. The next step in the synthesis of palmitate in the cytosol is simply the combination of this malonyl ACP and acetyl co-ACP that result in the production of butyryl ACP. We have discussed up to the production of 4-carbon fatty acid. Now this 4-carbon fatty acid then combine again with the malonyl ACP that result in a production of 6-carbon caproyl ACP. Then caproyl ACP is then combined with another malonyl ACP, result in the production of 8-carbon fatty acid. 8-carbon fatty acid then again react with the malonyl ACP or combined with the malonyl ACP, result in the production of 10-carbon and then 12, 14 and finally the 16-carbon palmityl ACP. So this is how a 16-carbon palmityl or palmitic acid is produced through the combination of acetyl ACP and malonyl ACP at the starting point. So as a summary, if we look at the complete reaction, there is a palmitate production where acetyl CoA, that's a 2-carbon fatty acid that combined with 7 malonyl CoA and with 14 NADPH as a source of energy or reducing equivalents, that result in the production of 16-carbon palmitate and 7 carbon dioxide, 14 NADP and 6 water molecule and 8 coenzymes A. So this is the overall reaction and the summary of this palmitate synthesis from the starting point of 2-carbon acetyl CoA. So if we look at the energetic efficiency of this system, so 8 acetyl CoA they are utilized during this reaction, and 7 ATPs they are utilized in this reaction, as well as there is a utilization of 14 NADPH or 14 NADP reducing equivalents. So that result in a production of 16 carbon fatty acid with the utilization of this amount of energy. Now come to the second system, that is simply the chain elongation. So in the second system, this is almost similar if we look at the first system, where there is a production of palmitate from acetyl-CoA by the addition of 2 carbon from the malonyl-CoA. So in the second system, that is considered as the chain elongation or the synthesis of long chain fatty acid, the palmityl-CoA combined with 2 carbon atom that result in the production of a stearic acid or stearate, that is an 18 carbon fatty acid. So this chain elongation steps, they require ATP and reducing equivalent in the form of NADPH. We are discussing the synthesis of fatty acid through chain elongation. In the previous slide, we have seen that this fatty acid, they are converted into long chain fatty acid through the addition of 2 carbon atom. So just keep in mind the incorporation of 2 carbon atom into the medium and long chain fatty acid, it requires ATP. Here you can see on the diagram at a first step where fatty acid is converted into fatty acyl coenzyme A, there is a requirement of 1 ATP. Similarly, during this these chain processes, in the next step that will be on the next slide, there is a requirement of 1 NADPH. So 1 ATP and 1 NADPH reduced NADP is required for this chain elongation process. During the previous slide, you have seen that the preferred substrate for chain elongation is the palmityl-CoA, and the product of this chain elongation is in most of the tissue is the stearic acid or stearate, that's an 18 carbon. So long chain saturated acid with 18, 20, 22 and 24 carbon atom, they are synthesized in the brain where they are required as a component of the brain lipid. And more important point is the mitochondrial system for elongation of fatty acid chains by using acetyl-CoA as the 2 carbon donor does exist, but it has a limited activity with acyl-CoA substrate with 16 or more carbon atom, and is probably concerned with the lengthening of the shorter chain. So during the chain elongation process, the fatty acid is converted into fatty acyl coenzyme A, then into keto acyl coenzyme A. So in the next this keto acyl coenzyme A is converted into beta hydroxy acyl coenzyme A, then into enoyl coenzyme A. In the next step, this enoyl coenzyme A is then converted into fatty acyl coenzyme A. So here you can see that there is again the requirement of NADPH or a reducing equivalent. So 1 ATP and 2 reducing equivalent in the form of NADPH, they are used during this chain elongation process or the conversion of this palmitate or 16 into 18 carbon fatty acid. The synthesis of fatty acid through the third system, that is through desaturation of preformed fatty acid. So in this system, the double bond, they are introduced into a fatty acid chain by the action of an enzyme, that is fatty acyl CoA desaturase. So these enzymes, they are present in the endoplasmic reticulum, and that result in a production of palmitoleic and oleic acid. So these two acids, they are produced from the corresponding saturated acid by a 9-desaturase system, stearoyl CoA desaturase, and these enzymes, they introduce a double bond between carbon atom number 9 and 10. The limitation of this system is confined to acid with a chain length of 15 or greater. It means that this double bond cannot be introduced if the chain length is less than this. Another important feature or thing which is related to the mammalian animal. So in case of mammalian animals or mammalian tissue, they have this delta 6 and delta 5 desaturases, but they do not have the system capable of introducing double bond beyond carbon atom number 9. So as a result, the mammalian tissues are not capable of synthesizing either the linoleic or alpha linolenic acid. Linoleic acid is a 18 carbon fatty acid that have 2 double bond. The one has position number 9 and the second as 12, whereas alpha linolenic acid is also a unsaturated fatty acid with 3 double bond. The first is at carbon number 9, then 12 and 15. So these fatty acids, they are considered as essential fatty acid in case of mammalian animals, and they should be provided in the diet. The reason being, they are not capable of synthesize these fatty acids. Once these essential fatty acids, they are provided in the diet, so they are capable of synthesizing a number of other fatty acids like gamma linolenic acid, arachidonic acid, eicosa pantoenoic acid and glucosa hexaenoic acid by successive chain elongation and delta 6 and or delta 5 desaturation activity. So this is the overall reaction of conversion of a saturated 16 carbon palmitic acid into a 16 carbon unsaturated fatty acid, where there is a double bond, that is palmito-oleate, and there is a requirement of reducing equivalent NaDH. So desaturation of this palmitate occurred, and that result in the formation of palmito-oleate. Then comes little bit the things which we have discussed in the previous slide already, that the mammalian cells, they contain delta 6 and delta 5 desaturations, but they are not capable of introducing the double bond beyond carbon atom number 9, so they are not able to synthesize the essential fatty acid like linoleic acid and linolenic acid. We have discussed in the previous slide already. Regulation of fatty acid synthesis. The regulation of fatty acid synthesis is controlled by enzymes, metabolites, end products, hormones and dietary manipulation. The first regulation is through this acetyl-CoA carboxylase. This enzyme controls the steps in fatty acid synthesis. Citrate promotes the polymer formation and favors fatty acid synthesis. Palmityl-CoA and malonyl-CoA cause depolymerization of the enzyme, and so they inhibit the fatty acid synthesis. The second is hormonal influence on acetyl-CoA carboxylase. Hormone glucagon, epinephrine and norepinephrine inactivate the enzyme by cyclic AMP dependent phosphorylation, so they inhibit the fatty acid synthesis. Whereas on the other hand, the insulin, it promotes fatty acid synthesis by dephosphorylation. The third is through dietary regulation, like the consumption of a high carb diet and a diet which is free from fatty acid, or you can say the fat free diet. It increases the synthesis of enzyme acetyl-CoA carboxylase and fatty acid synthase. Fasting and high fat diet decreases the synthesis of these two enzymes, and so decreases the fatty acid synthesis. So this is how this fatty acid synthesis is regulated in the body, and particularly it is regulated through the regulation of acetyl-CoA carboxylase, and that have under the influence of hormone as well as the intake of high carb diet or high fat diet. Thank you for watching.